And so today's just primarily going to be how you can design an experiment. Maybe by tomorrow, then we can now go properly to the workflow and the application of the CRISPR-Cas9. Then another important thing that would teach us tomorrow is the ethical concern, because when you just like what somebody said the other time, that whenever he hears the word gene editing, what comes to his mind is genetic modified organisms. So in as much as you're trying to learn in as much as you're trying to learn gene editing, it is very much important to be ethically grounded. So it's not just about the knowledge. So tomorrow is going to be about the applications and the ethical concern around gene editing. I was thinking I'm going to do the design and the workflow today, but due to one or two that is going on over here, I won't be able to do that. So before I proceed, the terms we need to get familiar with. I know most of us might probably know all of this and we are familiar with those terms, but for the sake of other people, so I want us to just go through them one by one. Of course, we know what gene is, which is a section of the DNA coding for a protein. Like I would always use an example. When you go to your university is your DNA. Each department, or would I say each faculty now, is a specific gene. So you have one DNA in your body. Let me say in this case now we have one DNA. Of course, it's not just only one DNA we have in the body. We have mitochondrial DNA, we have chromosomal or genomic DNA. So in this case now let's focus on genomic DNA. So let's say your university now is the genomic DNA, which is just one. Your gene is many. So your gene is more or less like a specific faculty, a specific segment of your university. Then we have what is called the exon. The exon is the coding region that remains in your mRNA. Of course, we should know what mRNA means. By this time, it's called messenger RNA, which is the type of the RNA we have. So we have intron as well. Why I'm trying to explain all of these things is that whenever you want to do your CRISPR-Cas9 study, all of these things are very, very important. So whenever I'm mentioning them, you know what it means. We have the introns. The introns are the non-coding region, which is spliced out of your mRNA. So they are non-coding. They are not going to be translated to a protein. So you have your untranslated region. The untranslated region, we call them UTR. So it can be 5 UTR, it can be 3 UTR. So the 5 UTR starts this. You can find your 5 UTR before your start codon. I repeat again, you can find your 5 UTR before your start codon. Then you can find your 3 UTR immediately after your stop codon. So we have the terminator, which signals the end of transcription. Of course, we know what transcription is, which is which your DNA is getting converted to RNA with an enzyme called transcriptase. So we have an upstream. Upstream just means a region before the 5 prime or a reference point. When I'm saying upstream my target site, it means before I get to my target site, the region before that target site, the 5 prime region before that target site is my upstream. Then we have a downstream. The downstream in the 5 prime region, which is like the down sequence after my target site, is my downstream. Then you have something we called homology hands. Remember yesterday we were talking about homology recombination or homology direct repair. So this guy is where you get to need them. Like I was telling us yesterday, the homology means, okay, if I should ask someone now, just a quick one, what is the meaning of homology? I think I made mention of it yesterday, so I just want us to know if we're following. So quick one, please, just one person. What is the meaning of homology?
Homology means belonging to the same, probably the same family, the same background, like the same story.
Thank you, Abimbola. I want to believe it is not only Abimbola that is on this call. However, Abimbola, get to message me after this program. It seems you are worthy of.
Right, sir.
So, okay, so that's a very beautiful answer. So we have the homology arms, which can be 5 homology hands, or three homology arms. So what it refers to as we can call them 5HR or 3HR. When we are designing our, maybe today or tomorrow, our donor template you're going to be listening to if I'm talking about 5HR, so you should know that I'm talking about homology hands. So by derivation, they are the DNA sequence matching a region around your cut site. Remember, we say you're going to give a donor template. Okay. So when you're knocking out a particular gene, which I'm going to show us soon, when you're knocking out a particular gene, the gene when you're knocking out a particular gene, when you're creating your donor template, it has to be similar to the gene you're knocking out. Maybe when I show us, we understand that part very well. It's not something I can explain with just mouse, and I have to show us. So we have a donor template, which I just talked about now. It is a DNA provided for a precise knock-in during your HDR, which is homology directed repair. That means you want to subject that particular template. Oh, this is what I want to put in after I've knocked a particular gene out. And the next one was going to be talking about the open reading frame. We have open reading frame, uh, which is a continuous stretch of a codon that starts from a start codon to your stop codon. So of course we know our stop codon, we know our start codon to be ATG, and we know our stop codon to be 3. Either you have your TAA, you have your TAG, you have your TGA. Then we have the same strand. So the same strand, we call them coding strand. When you have, what I call it, if you download your sequence, especially they are always in the same strand. So it adds the same sequence as your mRNA, except your T, which is to you in the RNA, and we have your antisense strand. Antisense is more or less the opposite of your sense strand. So it's a complement of the sense strand, and it is a strand that RNA polymerase reads to make your RNA. Then we have your plasmid, which I think I made mention of yesterday. The plasmid is a vector. The plasmid is a vector which helps you to carry your your or your gene of interest into the organism, into the whatever organism you want to introduce your gene is. So it's just like a carrier. That is just what it does. So it's a circular DNA vector for delivering your Cas9 and your guide RNA into the organism. Then we have your Cas9, which I don't want to talk about again because we've talked about them yesterday. Is an enzyme which is responsible for cutting. We call them the molecular scissors. It cuts DNA at a particular site with the help of the guide RNA. And we said the guide RNA direct the Cas9 to target the DNA. And then lastly, we have the PAM, which I've talked about yesterday as well, which is the short sequence that required for Cas9 binding. And I said the Cas9 the PAM can be NGG, it's NGG, and the N can be any nucleotide. So we can be TGG, AGG, CGG, or GGG. So that being said, so I'm just going to walk us through. And the first thing you need to do when you want to design your guide RNA, you want to design your CRISPR-Cas9 experiments, is to first, and I want you to be taking notes as I'm saying all of this. Most of them I know have them in the slide. Trust me. So I might be saying something that are not. Of course, this is my last slide. Maybe until tomorrow again. So you might not get to see. So it's better for you to take note. If you have your pen with you, just be picking those points down. So if you want to design your experiment, the first thing you need to do is to know the gene you're targeting. Just the same way you want to design your primers, of course, you have to know your gene. So if you know your gene, that helps you a lot. Then the next thing for you to do is to go to a database. So the first one, sorry, I'm not having all of those information here. Time doesn't permit me to put them together. Maybe by tomorrow, I will try as much as possible to put them together. So you have your database, you know your gene you want to target. Then after locating your gene, you're going to the database. So you can, of course, it depends on the particular gene you're working on. But for general purpose sake, the database we're going to be using is NCBI. So let me click on NCBI. Sorry, please, are we together? Are we together?
Yes, sir.
All right. So, like I said again, the CRISPR-Cas9 experiment is not just about the wet lab alone. So you have to be at least good to an extent in the bioinformatics part. Remember, the sources of your experiment start from the computational aspects. But whatever you design is what you're going to synthesize. So if your experiment is already wrong at the computational stage, then your wet lab experiment is going to be messed up. It's going to be messed up. So you have to be very, very careful when designing your experiments. Okay. So uh, can anybody suggest a gene, or maybe to not cause a while? And now let me just pick a random gene. Um, or do anybody wants to suggest a gene? Is anybody suggesting a gene? Anyone?
Sir.
B10.
GFP.
Say that again.
GFP.
So what is GFP? Let me ask you.
Uh, Green fluorescent protein.
And what does it do? What does it code? What protein does it code for? Or what is the function?
It's mainly used in lab to for easily editing work.
So it's mainly used in the lab for what?
Used in the lab for easily see if editing worked.
If editing work. So why do you now want to target the gene? Okay, maybe for context, the gene I'm saying we should name or we should suggest is a gene we want to target. So maybe I should just, for easy explanation so that we don't get confused, let's just use, of course, all of us know what sickle cell is, right?
Yes, sir.
Let's just use a sickle cell gene. So it's called the HBB. So you can take note of that, sickle cell HBB. So I believe the recording is going to be shared so that, or if perhaps the recording is not going to be shared, you can take screenshots or whatever you want to do, just do something to get an information. Okay, so we're doing Homo sapien, HBB. Oh, sorry. So we click on the gene because what we are trying to do here now, we're trying to download the sequence, the DNA sequence. Please, are we still together now? Like I said, today is going to be, I don't know, we are doing this physically. I would have been saying the way I'm doing it, I would say all of us should be doing it together. But if I'm saying we should do that, number one, I'm time limited as I am. And number two, is going to cause a whole lot of traffic on the call. However, if you can be following me the way I'm doing it, it's fine. If you want to do it when I'm done, all is fine. The most important thing is that you get the idea. This is just, I'm not expecting you to be as perfect today or perfect. No, it's just to give you the background knowledge so that you can develop it. And then, if you're now interested in gene editing research, I mean, you can take it up from there. So, please, are we together now?
So we can't see your screen, sir.
Away. What can you see, please?
We are still at the terms to get familiar with. That's what we are
Oh,
saying.
okay. Just give me a minute. Give me a second. Let me just correct that, please.
Okay.
Just some second, please. I'm so sorry. I'm trying to solve a technical issue from my side now. So sorry for that. Just to keep out so that it doesn't. Um, so, have we done any training on bioinformatics yet?
No, sir.
No, sir.
Okay. Okay. So I just want to be sure. So the first thing like I said, please just be, if you don't have your laptop with you or you can't do it, just be taking notes and probably practice it when you're free. You want to design a guide that there is no CRISPR-Cas9 experiments. As a matter of fact, that is the most important thing at this moment. In the chat box now. So if you know it, you can help us to drop it. I'm still waiting for an answer. Okay. Okay. Thank you. So let's continue now. Olamide said glutamic acid is being replaced by valine in position 6. Thank you for that. Okay, so what's this? Okay, so I clicked the gene, then it brought me to this interface. So I want to download the sequence. So I clicked on download, then it's going to ask me how do I want to download it now. So is it by transcript sequence? Is it by protein sequence? Is it by gene sequence? What I need for now is my gene sequence because your CRISPR works by gene sequence, not by your protein sequence. So I'm going to click on, is that light? Okay, so I'm going to click on gene sequence and then I'm going to download. Please, are we following? If you're
Yes,
following, just
sir.
okay, thank you. So I'm going to download. So we can see the download file here now. It comes in the zip, then open. Sorry, I'm being fast. I'm trying to beat time. But I promise tomorrow, even if you don't get it today, I don't just want to cancel the class. I told Miss Christiana, I don't just want to disappoint by canceling the class. So that's why I just have to come. However, I really need to be as fast as I can in order to meet up with my appointment. But tomorrow, we are there for each other, so we can spend as much as time we can tomorrow. Okay. So go to my just to open my file, and this is the sequence, the gene sequence. So can we see this? Can we see the sequence now? And see the sequence now.
Yes, sir. We can see the sequence.
Oh, thank you. Again, please, you have to just bear with me for today. I have less than 15 minutes to round it up. But I will try as much as possible to brief it tomorrow. Don't worry. We are there for each other. Please, just bear with me. So, I'm interested in this first sequence. I think the sequence downloaded 2 times. So just copy this first one. So I copy the first one, then open my Word document. Oh, no, 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 no. So then I open my Word document, then I'm going to paste it. Please, are we still together at this point?
Yes, sir.
All right, thank you. So I do something which is very personal. It is not a standard. It's just something me I love. You can see that it's not well justified. So there is a particular font I use that makes it to be well justified, and the font is Korea New. So it makes everything to be justified for me, so that it doesn't look scattered. Like I said, you can do your own manual justification, but it can be prone to error. You might unintentionally introduce a mutation, because once a particular letter is lost, the gene is already disrupted. That means you have even cut your gene without you using Cas9. All right. So the next thing that we need to do after you arrive at this place, remember I talked about UTR the other time, which is the 5 UTR and the 3 UTR to just carry us along. Where I think I made mention of where you can find your 5 UTR. Quickly, who can tell me where you can find your 5 UTR?
Before the start codon.
All right, thank you. And what is your start codon?
ATG.
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 Thank you. So the next thing I want to do is to identify my start codon and my stop codon. So once I can identify my start codon and my stop codon, it makes it easier for me to tell where my 5 UTR and my 3 UTR will be. So in doing that, I will go to this download again. This time around, you can see here. Can you see 5 UTR, 3 UTR here? Can we see? I might need audio response because I'm not on the chat
Yes,
now.
sir.
All right.
Yes.
So this time around, I'm going to download both the 5 UTR and the 3 UTR. Then I click 705. Wow. Then I go to the what I have downloaded now. Then this is my 5 UTR. So now listen, this is what I'm going to do. Remember, the 5 UTR is already part of my sequence here, but I really want to know where it is in the sequence. So that's why I'm downloading the 5 UTR. Everything is here. The 5 UTR, the 3 UTR, the exon, the intron, everything is here because this is a complete gene sequence. But I want to to just identify where my 5 UTR is. That is why I downloaded the 5 UTR and the 3 UTR separately. So this is what I'm going to do. I'm going to copy. This is for my 5 UTR now. Copy my 5 UTR, then find it. So just find it inside the sequence. So can we see it, please?
Yes, sir.
So this
Please.
means this is my start codon, which is ATG. So the next thing I will do is to maybe color the ATG maybe with the green, and color this one maybe with a color that, or the best thing I do for my 5 UT for my UTR is to turn them to small letters. It's just all these things is just to make sure your work are neat and you are not, everything is not looking the same, it's not scatter. So I turn my 5 UTR into a lower case because they're not coding, they are not a coding sequence. So here now I can say this is my start codon. Then I do the same thing. Please, do we understand what I did here now?
Yes, please.
Oh, thank you. So then I do the same thing for my 3 UTR. Sorry, 707. Wow, time is really, really fast. So I do the same thing for my 3 UTR. 3 UTR is kind of long, so I may not be able to copy everything. I just copy because what I just need to see is the beginning of the 3 UTR in order for me to, what I'm trying to achieve there is where is my stop codon. So let me just copy the first nucleotide. Because I might copy everything, I might not be able to search for everything. So let me just copy the ones you can search. Then I go back to my Word, then search. So can we see now? So it means everything. Oh, sorry. It means everything here is my 3 UTR, 3 UTR. So I turn them to small letters as well. Then this is my stop codon, TAA. Then I can pin this one maybe red. Please, do we understand up to this point?
Yes, sir.
Yes, please.
All right. God help me. Okay, so now we know our start codon, we know our stop codon, we know our 5 UTR, we know our 3 UTR. The next thing we want to do is to, if you already have your target site, we're just going to use a 1 for, ideally, we ought to go to the codon 6, position 6. But because of the complexity, because if I want to do that, I'm going to look for an adjacent gene of this particular HBB, and that is going to make us to be it is a complex step. So I wouldn't want us to bore us with that or make it complicated for us. So if I'm going to target codon 6, meaning I have to get a gene before HBB, so it's going to be part of my 5 UTR, but that is a complex step. So we're just going to do would I say an hypothetical gene now, or an hypothetical target site. So let me just pick a particular site so that we can, for easier understanding. So let me say I want to pick. Let me look for a particular GTG. Okay, let me say I want to target a GTG. So let me say I want to target this particular region now. So let me label this site. So let me name it target site. Let me label the target site. Sorry, please, I have less than 5 minutes to go. I'm very, very sorry for this. If we can't continue this site this way, then we take it up again for tomorrow. But I promise you, tomorrow is going to be fun. So you just have to bear with me today, please. So now we have our target site. Now the next thing we are going to do is to now look for a guide RNA, design a guide RNA that is going to help us, that is going to be closer to our target site. That is the next thing we want to do now. We've identified 5HR, I'm sorry, the 5 UTR, the 3 UTR, the start codon, and the stop codon. Before you can say you want to do your 5HR and 3HR, your target site has to be known. In this case now, your 5HR has to now be upstream of your target site. So anything from here now, anything from here upstream of my target site is my 5HR. Anything after my target site is my 3HR. That's downstream. Please, do we understand?
Yes, sir.
All right. It's only one person that is talking. I just want to believe we all understand. However, don't worry. Tomorrow you can bombard me with questions. But for now, let's just flow, please. I'm really, really pressed for time. So now we've explained what 5HR and 3HR means. So the next thing we're now going to do is to go to a particular site called chop chop, not the chop chop of small chop. So so you can see, just press chop chop CRISPR. So it's going to take you there, because if you press online chop chop, they will be bringing you food for you. But press chop chop CRISPR. So this is it. Ideally, we have two sites we can use to design the guide RNA. Maybe I will just explain for chop chop now. Tomorrow I will explain the second site, the second option. Like I told us, CRISPR-Cas9 involve a lot of bioinformatics. So this is chop chop now. This is the interface. So the first thing I'm going to do, remember I want to design my guide RNA. So these are the things he will ask me first. Remember we are doing for human. So human is Homo sapiens, right? Yes. If you are doing for any other organism, you have to click it, be it Plasmodium, be it bacteria, whatever the bacteria is, the fungi, just click it. But now we are doing for human. So that's why it remains Homo sapien. We are using CRISPR-Cas9 because we have other ones. I made mention of, you can see Talent here now. I made mention of CRISPR-Cas13, you know, the different options you can use, but we are making use of CRISPR-Cas9. We want to do a knockout where this doesn't really matter. Well, these two matters a lot. Repression, like I told us yesterday, you are not trying to knock out the gene. You're just trying to repress the expression of that gene. So we're not doing that. So we just want to knock out so that we can introduce a new template. Then next thing is to click on paste target so that you will be able to now paste your sequence. So what you're going to do is to go back to your sequence now. Go back to your sequence. So let me see the region. So let me see the word counts here now. Okay, so my target site, because I copied right from my target site. My target size is around 753 base pair. That is my target site. So I'm going to copy all the sequence. I'm going to copy all the sequence. The location where I'm trying to target is 5753. So I copy all the sequence, then paste, then find target. So you allow it to load. Oh, sometimes it can take time, sometimes depending on your service and depending on how the server is busy because you're not the only one working. A lot of people are working at the same time. So you can see it's getting longer, as more than expected. So I guess the server is busy. Oh, this one is not open at all. Okay, so it's not loading at the moment now. I guess the server is very, very, very busy. A lot of people are working, and I'm already pressed for time. So, plug, do we understand up to this point, please? I just need the feedback.
Yes, sir.
Yes, sir. Yes, sir.
All right.
Yes, sir.
So, I might have to go now. However, tomorrow I just want to trust that everything will be fine tomorrow with the network. So, we will do. Would I say we have more time tomorrow? Okay. So I might probably repeat all of these things I've said so that we can just link it up with the applications and the ethical consent. So that will be what we're going to be doing tomorrow. I plan to finish this up, but okay, okay. So let's just finish this one so that we can go. So can we see this interface now?
Yes, sir.
All right. So this is what is going to bring for you different guide RNA, as you can see. Remember I talked about Palm site yesterday, so you can see all of them as the Palm site. This is TGG, 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 AGG, and like that, like that. So if I may ask, what is the location of our target site? 753. I made mention, right?
Yes, sir.
So you have to. These are the things you need to consider before picking your guide RNA. So we won't pick our guide RNA today. I will just explain all of these criteria. So by tomorrow, then we'll pick our guide RNA. I'm already out of time. So this is rank. 1, 2, 3. This is the target sequence. This is I mean, we have how many guide RNA here now? In just that sequence, we have 164 guide RNA. So this is the genomic location, which guides you on where to target. Is your sequence around 1458 base pair? Is it around 602? It gives you that region. Okay, this is that's why I say you need to know your target site. So it helps you to locate whichever one you want to do. So this is your strand. Is your guide RNA present on the same strand or is it present on the antisense strand? This minus means antisense strand. This plus means on the same strand. Please, do we get that?
Yes, sir.
Right, thank you. Then just like your primer, you have your GC content. 40 to 60 is ideal. Then you have your self-complementarity. Self-complementarity means if your guide RNA is not going to bind to itself, just like your primer, which can lead to primer dimer. So you have to be sure that your self-complementarity is at least 0. Then other criteria you now need to check is your MMO, which is mismatch potential. Mismatch 0, 1, 2, and 3. So when you have one here, it's still actually fine, but the others should be at least, depend. Although sometimes you might not really get what you're looking. You might not really get everything to be 0, 0, 3. Just look for the best one you can work with. Of course, you can still optimize your experiments. At least it minimize, like I said yesterday, CRISPR-Cas9 is not an 100% kind of experiment. If the success is always low, so you just have to get yourself to being bullied by the experiments so that you don't keep having outbreaks. So then last one is your efficiency. How efficient is your guide RNA? So now this is the balance. You can't, because your efficiency is high, we now judge with that. No. So all these things has to be put in place. The first thing is the most special and most important thing is your genomic location, especially if you want to do precise mutagenesis. If only you're trying to do conditional knockdown, maybe tomorrow I will explain that. You might not really be concerned much about that. But if your objective is a precise mutagenesis, genomic location is your best bet. Then you check that. Then you check if your strand is on the same strand on the antisense strand. Check your GC content. Check your self-complementarity, check all of the mismatch potential, then check efficiency. As a matter of fact, you might even have an efficiency which is 40 something or 50 something. So far, these ones are good. Then you can still, so that's what I'm saying. You know how to balance your experiments. You might not necessarily have the most perfect one, of course, no, because I mean they are genes, so you are not the one that designed the genes. You're only trying to design a guide RNA you can use. So look for how you can pick the one that is best in respect to your location that you are good to go. Please, do we understand up to this point?
Yes,
Yes, thank you.
yes, sir.
Like I said, I'm already out of time already. So please, let's stop here for now. So tomorrow we are now going to, with all the explanation I've made, of course, somebody is going to give me a recap of the criteria. So we are now going to pick the guide RNA we'll be using, then continue from there. So, Miss Christiana, I'm sorry I have to go now.